Okay, this is the third part of lecture 10, and in this part, mainly I'll discuss how recombinant DNA technology can improve the production of alcohol or can address some of the problem associated with alcohol production. So we know to produce alcohol we use different raw materials. Basically, those raw materials are used as source of esters and sugars. So from the esters and sugars, alcohol is produced. Okay, so we already know that using fermentation we produce alcohol. So, for example, we can use grapes, uh, rice, malts, okay, to use it as raw materials, and by fermentation using microorganism you can convert them into alcohol or produce alcohol. Okay, however, there are different raw materials which you can use commercially. So for example, milled grain, okay, mild grain are used to produce alcohol. But although we are saying that we are using fermentation, but in many cases this is not a single step process. Okay, so before starting the fermentation, the raw materials need some pre-processing. Okay, so for example, for mild grain, when mild grain is used as raw material to produce alcohol via fermentation, before going to the fermentation some pre-processing is required. So for mild grain, first with they are treated with steam. Okay, and steam turn them into gel like form. Okay, so if mild grain is treated with steam, a gelatinized material is formed. It is actually necessary because when this gelatinized material is formed, the exposed surface area of the esters, okay, it increases. That means the uh, and that means after heat treatment, when the mild grain they turned into gelatinized material, the surface area of the esters get exposed, so they become more readily available for the subsequent enzymatic reactions. Okay, so after gelatinized material is formed, they need to be cooled down because steam is very hot. Okay, so in this, if the the gelatinous material is very hot and we add the further enzymes, those enzymes will get destroyed. Okay, so after gelatinization is formed by steam, it need to be cooled down to like around 50 to 60 degrees Celsius, and then alpha amylase is added. Alpha amylase liquefied it. Okay, so actually alpha amylase transform it into low molecular weight polysaccharide. Okay, and this reaction is need to be done at around a little bit higher temperature. Like we cannot use 100 degrees Celsius. If we could use 90 degrees Celsius, it would be better, but we cannot use that temperature because at that temperature alpha amylase will be destroyed. Okay, so 50 to 60 degrees is a kind of good temperature, and this temperature is important because otherwise the effectiveness of the enzyme get down. Okay, in the enzyme they cannot efficiently penetrate the gelatinized material. So if they cannot penetrate the gelatinized material, they will not be able to hydrolyze it and turn it into low molecular weight polysaccharides. That means liquefied them. Okay, then after this step, glucoamylase enzyme is used to saccharify them. Okay, and then from this saccharified material, either we can produce alcohol by fermentation using like yeast, or we can produce fructose by using another enzyme, glucose isomerase. Okay, so now in this process, okay, different enzymes are used. You can see like alpha amylase, glucoamylase. Okay, so these amounts are not cheap. Sorry, these enzymes are not cheap. Okay, so these are costly. Okay, so in commercial production, this enzyme adds some cost, and that cost is actually not a small, it's a huge cost. So this enzyme adds some cost in the process. Okay. Okay, and not only alcohol, I mean, a significant amount of commercially used fructose, which are used in the like for in the beverage or food, are produced from mild grain, especially from the corn. Okay, and those production require these enzymes, the process is costly. Okay, so now by using recombinant DNA technology, we can actually address this problem. How? So first we have to identify the those points which are increasing the cost. One I have already said, that this enzyme like the glucoamylase, alpha amylase or glucose isomerase, this enzyme itself is costly. Okay, then look at this process. First we use steam, then cool it down. Why we need to cool it down? Because alpha amylase will be destroyed if we add them in the steam hot temperature. Okay, so this step needs some extra cost, right? Because we have to cool it down, it will require a energy cost. Okay, then like after alpha amylase we use another enzyme, glucoamylase. So the condition required for optimum activity of these enzymes are not same, such as the pH or temperature requirement. Okay, so first we use alpha amylase, then actually we need to change the condition which are favorable for glucoamylase. Again some extra costs are added here. Okay, so now how we can solve these problems by using recombinant DNA technology. Okay, so we actually can do it by several way. So one is, if we can uh, by genetic, I mean the enzymes which are used in production of, I mean these enzymes, this itself are produced by fermentation technology. Okay, I mean the what I wanted to mean, the enzymes which are used in production of fructose or alcohol from the mild grain such as corn, uh, corn flour. Okay, are, I mean these enzymes itself are themselves are produced by fermentation process. So now if we can, by through genetic engineering, if we can made recombinant microorganisms which can produce high amount of this kind of enzyme, then the production cost of this enzyme will be lower. So not directly but indirectly, as these enzymes are used in this process, the production cost of this process will be lower indirectly, because we then will be able to get uh, enzymes in a cheaper price. Okay, so this is I mean this can be one solution. Another solution is, so in these two steps, first we use steam, then we have to cool it down to add alpha amylase. Now if we can't I mean if we can't make a heat tolerant alpha amylase, okay, if we can, by using genetic engineering, if we can produce a heat tolerant alpha amylase, then we don't need to cool it down. I mean after steam treatment we just can add the alpha amylase because it is heat tolerant. So if we can do this, I mean if we can produce a heat tolerant alpha amylase through recombinant technology, then again it will solve the problem. And also it is theoretically possible to produce a alpha amylase which has similar requirement for glucoamylase, or like using recombinant DNA technology, if we can make alpha amylase or glucoamylase which have similar requirement for the conditions, right? Like pH requirement, pressure requirement, temperature requirement for the optimal activity. Okay, if both these enzymes work in same conditions, again it will help to lower the cost. Okay, so these are the strategies which by which we can solve the problem. What problem? Which add the production cost. Okay, so by, okay, so we can, as we can will be able to address this problem, we will be able to reduce the production cost. Okay, so well, indeed actually different, I mean scientists there trying to uh, okay, so there is another strategy to address the problem, such as For example, um, if we can engineer an enzyme, okay, which can efficiently degrade raw esters. Okay, so what that mean? I mean, a mild grind is the raw esters. Okay, so if we, as I mean, in this process, gelatinization is important. I mean, from mild grind, we first have to do the gelatinization, then do the further enzymatic process. But if we can engineer an enzyme which will which don't need gelatinization, I mean, which can directly transform the mild grind into liquefied material, then again it will help to reduce the cost. Okay, so anyway, these are some recombinant. I mean, these are some potential approach that we can do through recombinant DNA technology, and if we are able to do that, then we will be able to reduce the cost. Okay. So well, I mean, although this sounds very cute, I mean, this sounds that theoretically these are very possible, but in a real world, I mean, these are not that simple always. Okay, so I mean, when we really go for research, various problems occurs. Okay, sometimes the experiment doesn't work just okay, and you will not be able to find out why the experiments are not working. But anyway, there will be always problem. It doesn't mean that we researchers will not do the research. Okay, so many scientists around the globe, they're actually trying to they're using I mean they're trying to utilize the recombinant DNA technology to achieve these approaches to solve the problems associated with the production, alcohol production, when we utilize raw materials like uh, raw raw materials like corn. Okay. Okay, so what actually so the scientists are trying to do? So in one attempt to develop or isolate a alpha amylase, and alpha amylase is involved in this step. Okay, so scientists, they're trying to identify or isolate a heat tolerant or heat stable alpha amylase. Okay, so how they're trying to do that? Like for example, Bacillus stereothermophilus is a heat tolerant bacteria, and it produces alpha amylase which is heat tolerant. Okay, and so scientists, they have isolated the gene for alpha amylase from this heat tolerant bacteria and clone them or transform them into Bacillus subtilis, this bacteria. Okay, so this bacteria, now this recombinant bacteria produce the alpha amylase which is heat tolerant. Okay. Okay, now then actually okay, so and in another approach, scientists were able to develop a recombinant Saccharomyces cerevisiae strain which can produce glucoamylase enzyme. Okay, so if we use this recombinant Saccharomyces cerevisiae, then we will be able to skip this step. Okay, so basically, I mean, yeast, Saccharomyces cerevisiae is a yeast, right? And this yeast is used to produce alcohol from saccharified material. But in that approach, scientists were able to produce a recombinant yeast which is able to directly use liquefied material. Okay, it don't require the saccharified material. It can directly use liquefied material because that recombinant yeast has the gene to synthesize this enzyme, glucoamylase. So as this recombinant yeast, they can synthesize the glucoamylase uh, enzyme. They can directly use the liquefied material. Thus, if we are able to use this recombinant yeast, then we will be able to skip this step, which will help to reduce the cost, production cost of alcohol. Okay, so how actually they did that? I mean, how they develop this recombinant yeast? Well, they have isolated a gene which encode the glucoamylase from Aspergillus amuri, okay, from this fungus, and then they clone this Aspergillus amuri gene. Then they clone this gene, glucoamylase gene, into the Saccharomyces cerevisiae. Okay. Now, well, again, it sounds very cute, but this strategy also have problem. The problem is, the Saccharomyces cerevisiae strain, okay, they cannot tolerate high level of alcohol. Okay, so if the level of alcohol is high in the fermentation, in the fermenter, then their efficiency gets reduced greatly. Okay, and another problem is this recombinant Saccharomyces cerevisiae, they don't produce good amount of glucoamylase. The gene for glucoamylase were introduced into them, but they, although they have the gene, they don't produce much amount of glucoamylase. Okay, and there is another problem. This gene for the glucoamylase was introduced into the Saccharomyces cerevisiae by plasmid. Okay, and this recombinant cerevisiae, they tend to lose this plasmid. So if they lost this plasmid, well, they will not produce glucoamylase at all. Okay. Well, although there are problems, no problems is insurmountable. Theoretically, every problem is solvable through science. Okay, so actually, scientists are that they're trying to solve this, address this problem, and some of the problems have already been solved. Okay, so like uh, one problem, like this recombinant cerevisiae, they produce low amount of amylase. It was resolved by by deleting some negative regulatory region, okay, from the plasmid. So the plasmid which contain the gene for glucoamylase, okay, it have some regulatory region, okay, and some of the regulatory region, they affect the expression negatively. So after deleting those negative regulatory region, the production of glucoamylase was enhanced. Okay, and another approach was, I mean, they integrate the plasmid into the genome of the Saccharomyces cerevisiae. So thus, as now they are integrated into the genome of the Saccharomyces cerevisiae, there is no chance to lose it. Okay. Okay. And another solution was uh, new strain of Saccharomyces cerevisiae. Scientists were able to isolate a better alcohol tolerant Saccharomyces cerevisiae strain. Okay, and use it to make the recombinant strain. Okay, so by these approaches, these problems associated with the recombinant Saccharomyces cerevisiae were resolved. Okay, so that was about the use of, the recombinant DNA technology for the production of fructose and alcohol using raw materials like mild grain. Okay. Okay, so let's talk about alcohol little bit more. So uh, low ethanol wines. So what low ethanol wines? So many wine drinkers, they prefer that the amount of alcohol in their drinks will be lower, but it will be rich in flavor. Okay, so that means the wines will be rich, the wine should have intense flavor, very good flavor, high flavor, but the amount of alcohol should be low. Okay, however, the usual strain which are used to produce alcohol, basically if amount of sugar is high in the raw materials, more alcohol is produced by those strain. Okay, so that means what will be the final amount of alcohol? Final amount of alcohol in the product, it depends on the amount of sugar in the raw material. Okay, So, and good wines are actually produced from matured grapes. Okay, and matured grapes juice, they contain high amount of sugar. So by using the traditional or usual yeast strain, it is difficult to produce this kind of alcohol, which has low amount of ethanol in it. Okay, so however, uh, scientists are trying to generate engineered yeast. Okay, engineered brewing yeast, which which produce low amount of alcohol from grape juice. Okay, so if this kind of strain is, we can develop this kind of strain, genetically engineered used strain which produce low amount of alcohol from sugar rich grape juice, then we will be able to produce this kind of things. Okay. Okay, it may sounds that we want more alcohol. Why we want to produce low amount of alcohol? It's the public demand, right? So wine are produced not for commercial purposes. They are produced for drinks. I mean, not for commercial means not for industrial use. They are produced to use as beverages. Okay. 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 Now let's try to learn about use of recombinant bacteria microbes in Silas fermentation. So what is Silas fermentation? Okay. Uh, Many crops such as grasses, corn, alfalfa, they are used as animal feed. Okay, so they need to be preserved after the crop is harvested. Okay, so you know that like say for example খড় যে ধান মাড়াই করার পর যে খড় থাকে okay, so after you harvest the crops, so with the remaining they can be used like grasses or even like the corn itself, alfalfa, they can be used as a animal feed, so they need to be preserved for long term. Okay, so if we just left them, different microbes will attack them and they will be get degraded. Okay, so how the preservation can be done? One way is by adding lactic acid producing bacteria into them. So okay, so the lactic producing bacteria, they produce lactic acid, and this lactic acid reduce the low pH, and at low pH most of the other spoilers organisms, they cannot grow. Okay, so by this way we can preserve these crops. Okay, so this is called silage. Okay, and actually in this silage those lactic acid producing bacteria will grow. Okay, that means it's a fermentation process, so that's why it is called silage fermentation. Okay. Okay, now this has some problem. What problem? One, I mean, these crops, okay, they usually don't contain high amount of lactic acid producing bacteria, so we need to use lactic acid, inoculum of lactic acid bacteria, say typically L plantarum. Okay, lactobacillus plantarum, this bacteria is used to preserve this crop. It's a lactic acid producing bacteria. Okay, so as the initial fresh uh, crops, they don't contain uh, good numbers or amount of the bacteria, we need to add this bacteria, inoculate the bacteria. Okay, so that means we actually have to produce this bacteria by other fermentation. Okay, anyway, it was one problem. Another problem is if these crops, they contain low amount of water soluble carbohydrates, then these bacteria, lactobacillus plantarum, they cannot grow efficiently on them. Okay, so that means if the crops have low amount of water soluble carbohydrates, this is not favorable for the growth of lactic acid producing bacteria. Okay, so now how we can address this problem, how we can solve this problem? Again, we can take the help from genetic engineering, recombinant DNA technology. Okay, so one approach was a recombinant L plantarum, lactobacillus plantarum. Okay, a recombinant lactobacillus plantarum strain uh, can be developed. Okay, actually the scientists are trying to develop this strain. Uh, so how we can develop this strain? Say a gene uh, was isolated. Which gene? A gene for alpha amylase gene was isolated from Lactobacillus amylovirus bacteria, and then it was integrated into the chromosome of the L palanterum. Now this recombinant bacteria, it can utilize carbohydrate, even those carbohydrates are not that much water soluble in the crops. This recombinant bacteria, they can utilize that carbohydrate. Okay, so this will actually solve the second problem. Okay, so this is one approach, okay, to improve the silage fermentation process. Okay, so basically that's all for this lecture. Okay, so I mean, if you go through the rest of the slides, you will find that some you will find that utilization, I mean, utilization of other raw materials such as cellulose, lignins, hemicellulose, these have been discussed. Okay, so I mean how these are used and how recombinant technology can help to improve the utilization of these compounds. Okay, so these kind of things have been discussed in the rest of the slides. Okay, so I will suggest that just go through this slide and it will not be that difficult to understand. Okay, so that's all for lecture 10. Thank you for your patience.